So this video was originally created back in 2014, and so it's been 10 years since it was actually updated. Interestingly, when I started updating this video, I found that the concepts within them haven't changed that much, which, to be honest, isn't surprising, because the underlying chemistry hasn't changed at all. So I'd like to spend the next 10 or 15 minutes discussing how to get proteins out of samples and how to analyze them. So at the beginning, I just want to emphasize that this is a general guide, okay? Every sample that you deal with is going to be different, and there's not a universal method, no matter what the literature actually tells you. So with this in mind, you've got two choices. Your first is to tweak your method for every sample that you have to enhance your data quality. This takes time, so you're going to be spending a lot of time optimizing each different condition to see whether that actually has an effect on the data that you generate, the depth of the proteome that you extract, and the effect on data quality and reproducibility. The other alternative is just have your method, use it for everything, and get work done so that you can get your publications out and you can achieve your goals. This might not get everything out of the sample, and that has to be acknowledged and reported when you're writing your manuscripts to say that, look, I might have missed something because I haven't extracted it. There's nothing wrong with that. So what you need to do is that you need to determine your study objectives and you need to plan your experiments accordingly. So before we start smashing things apart, we need to remember what proteomics is actually trying to do and how we actually analyze proteoforms. So I'm going to go back to a Lego analogy that I started with a few years ago. And so with this particular Lego kit, and here's the box, you can actually make these four really cool different models out of this one Lego kit. And so what you can do is you can say that the parts within the Lego kit are, in the case of a proteome, are actually your amino acids, your building blocks of your different proteins. And then the instructions within the box can be considered your genome, which is the blueprint that tells you how a particular proteoform actually goes together. So what we're doing in proteomics is we're not only identifying which particular proteoforms are present in the sample, what particular things have been made by translating the genome, but we're also looking at quantifying the amount of a particular proteoform that actually is in the sample. How many molecules of exactly the same thing are there in our sample? So how does a cell actually change the number of these proteoforms? Well, the simplest way that it can do that is that if it needs to respond quickly to an environmental change, it can just make more machines. Okay, so in this particular case, we've got our this little skid steer here. And so if I want to dig more holes more quickly, I can just make more skid steers. I can just bring them in and actually get the job done that much quicker. And the proteome is the similar kind of thing. The cell goes, okay, wait a minute. Here's this stimulus. I need to do this more quickly. I make more machines. That happens more quickly. The other way that we can do it is we can actually change the machine. So we can change the proteoform to a different proteoform. So in the analogy of our skid steer here, it's basically the same as putting different attachments here on the front of our skid steer to achieve different tasks. And we can do that with proteoforms that are already existing in the sample. So these proteoforms, they can undergo proteolytic cleavage to make smaller proteoforms that have a different function. You can post-translationally modify your proteoforms by phosphorylating them or acetylating them. That changes the chemical nature of them and can change the activity on the proteoform. Or you can assemble proteoforms into protein complexes to get them to do different jobs that the individual proteoforms couldn't do before. So what we aim to do in proteomics is we want to isolate all of the molecules of a particular proteoform to analyze them together. So here what I've done is I've taken our different models here and I've represented them as these colored balls. And what I really want to get across here in this slide is that when we talk about a protein or we talk about a proteoform, we're not talking about one single molecule. There's not one single molecule of a protein in a cell or a proteoform in a cell. There are hundreds, there are potentially thousands, tens of thousands of molecules of exactly the same thing that defines a proteoform. So in this particular case here, where our proteoform is represented by a triangle, when we add a post-translational modification like phosphorylation, we're creating a completely new proteoform. And that has different properties to the old proteoform. And what we want to try and do is resolve these unmodified proteoforms away from the modified proteoforms to be able to better analyze them. Now, this bucket is representative of our cell. And so everything is mixed together within the cell. All of these proteoform molecules are floating around all together, and we need to separate them out. So what we ultimately want to do is we want to get all of the molecules of the same proteoform in the same place. Now, whether that's actually putting them into their own bucket or their own tube or resolving them into their own single band or spot on an electrophoresis gel, or getting them to be in the same chromatographic peak, it's all the same thing essentially. So we're going to talk about some basic biological chemistry. So the majority of organisms that exist, I dare you to find one that doesn't, are made up mainly of water. And the biological molecules have to interact with water molecules to actually affect their activity to be able to perform their biological function. So the 20 amino acids that exist within a cell interact with water through hydrogen bonding and van der Waals forces and all of those kind of things. So what ends up happening is that these charged groups on the molecule can form hydrogen bonding with water. The other thing that can happen is that you get these secondary structures that are being formed either through hydrogen bonds or through hydrophobic interactions which exclude water. You can have chemical bonds like these disulfide bridges. You can also have these ionic bonds that occur. All of these either interacting within that background of water to form either complexes or interact with lipid membranes and things like that. So these things can then fold together in different amino acids. So this is basically showing the hydrophobic and the hydrophilic regions of the molecule, where the yellow beads are hydrophobic amino acids, and these can associate together to form these secondary structures. So proteins form these globular structures in water-based solutions where the charged amino acids or the hydrophilic amino acids are on the outside, and they are interacting with these water molecules represented by these balls where the red is the oxygen, the white is the hydrogens that are bonded onto that oxygen to make your water molecule. So you can see that this protein molecule is surrounded by water molecules. It'll also be surrounded by salt molecules and things like that that are interacting with the protein itself as well. So these can then form up into these secondary structures where you have the hydrophobic amino acids all associating together, and then the hydrophilic ones on the outside. This is a side view, this is a top view of what's going on in this protein association to make what is likely a transmembrane channel. And that then looks like this in a cartoon form, where you've got the hydrophobic region of the protein, this is the transmembrane domain, lining up with the hydrophobic tails of your lipids in your bilayer membrane and your hydrophilic head group of the lipid, typically a phospholipid. So unless the protein molecules, which again we're representing by these balls So each one of these balls represents different molecules of the same proteoform. Unless they can be floating around in liquid, you can't really separate them from other proteoforms. There's no way of getting them apart. And so you need to separate the proteoforms away from other proteoforms to reduce the complexity of the sample so that as many proteoforms as possible are being identified and quantified. Now, what ends up happening is that when you take these proteoforms out of their native environment, if they are just in water with no other buffer salts or no other molecules in there, they typically just fall out of solution. Okay, they fall out of solution and they form an aggregate at the bottom of your tube. And the problem with this is once they're aggregated, you can't separate them. You can't move them around in liquid to actually get them to separate. So what you then need to do, so this again is shown in the bilayer membrane. Within the membrane, these things are not able to be separated effectively. You need to get them out of the membrane. So, isolate them as, these monomers, somehow get them suspended in liquid. Now, these are hydrophobic molecules. They don't want to be suspended in liquid. So you've got to actually add something to them to help them to get out of the liquid, rather than have them aggregate and precipitate where you can't get them. So here's our particular cell, all right? And what we need to do is we need to bust this thing open, okay? Because we can't get to the proteins unless we break these things open. So you can originally keep cells intact to try and isolate the cells away from things like culture supernatant and things like that, which if you're working in cell lines and things like that is a really, really, really important thing that you need to do. You need to wash away all of those fetal calf serum contaminants, unless you basically want to profile the proteome of fetal calf serum, which no one wants to do. So what we need to do is we need to be able to break these things open. We need to break open the membrane to be able to release these soluble proteins on the inside, to be able to then separate them and analyze them. So when we do that, if we're just doing this in a basic PBS solution or something like that, break the cells open, we do a centrifugation step. The proteins embedded in these membranes are very, very large. They're very, very dense. You can centrifuge them down to the bottom of a tube and you end up with the soluble proteins in the top, and you end up with a mixture of all of the membranes and the associated membrane proteins inside those. So you can then take those and you can do various different separation types. So in this particular case, this is a really basic form of chromatography. Let's say that this is size exclusion chromatography, where the smaller proteins are migrating faster through, and you can collect all of those in a single fraction, collect the next larger protein in a single fraction, and so on and so forth. It's a very, very simple way of doing some crude fractionation. Will you get absolutely pure proteoforms by doing this? No. And then you've got to deal with this stuff in the bottom. So how do you deal with these insoluble proteins that are down here in the bottom? So the insoluble proteins here, what you need to do is you start to replace these the water molecules with chemical additives that actually enhance the solubility of the protein and get them to float around in solution. And there's a couple of different ones that you can do here. The first that we're going to explore here are detergents. And so the correct terminology is actually surfactant. A detergent is something that you wash your dishes in, but the theory is essentially the same. So detergents are surface active compounds, and so what they do is they consist typically of a long hydrophobic tail region and a hydrophilic head region. The tail can interact with hydrophobic lipids and hydrophobic regions of the protein, whereas the head group interacts with the water environment that's around it. So these things can disrupt the membrane. They can replace the lipids in the membrane, to be able to solubilize these proteins into solution. So, not quite sure why I've drawn that ball. But what you're doing here is that you've got these balls of protein that are insoluble, and therefore what you can do is you can start to disrupt the individual molecules of the proteoforms and coat them with these surfactants to be able to get all of the molecules suspended singularly in solution, so that you can then isolate all of the molecules of a particular proteoform to be able to analyze. The other way to do this is to use things like chaotropes, and chaotropes kind of fallen out of fashion a little bit with the focus more on shotgun peptide-centric proteomics. There is still a lot of people who use these to do extraction for shotgun work, but they're mainly found in doing things for two-dimensional gel electrophoresis, where you're doing isoelectric focusing before you do size separation. So the three main chaotropes that you've got here are the ones that we see here: urea, thiourea, and guanidine, and these are usually used in really, really high concentrations. So typically you'll see things where you've got 8 molar urea as the chaotrope in your extractions. The other one that you see a lot is 7 molar urea and 2 molar thiourea, which is a more chaotropic solution than urea by itself. So what these things do is they disrupt the hydrogen bonding of water with the surface of the protein, and so they're replacing these water molecules with molecules of the chaotrope, and they're allowing the protein to be opened up, the structure of the protein to be opened up to expose those hydrophobic regions and stabilize those so that they're more able to go into solution and therefore be separated. We just talked about surfactants just then. As I said, they're surface active reagents. They're typically water soluble because of this head group. This is SDS, which is the most common surfactant that the majority of people use. And what you see here is this long hydrophobic tail that can interact with hydrophobic regions of the protein, and this charged head group of the sulfonate. And so what these do is that the SDS molecules, you end up linearizing the protein either by boiling it or just having it in solution, and the hydrophobic tail region interacts with the hydrophobic regions of the protein and start coating the molecule. And as they do, they open up the structure of the protein molecule so that you can get more and more of these SDS molecules associating. And then you've got these negatively charged groups that are now interacting with water that surrounds this protein surfactant complex and allows that to be suspended in water. There's loads of different surfactants. SDS is one of the most popular. Anionic detergents are very popular. The other main anionic detergent that you will see a lot in the literature is sodium deoxycholate. That has become very, very popular because trypsin, which you use to create peptides from your proteins, is active in very high concentrations of deoxycholate, typically up to 1%, where SDS, you need to dilute it down to below 0.1% to be able to have the trypsin to be active. So the other kinds of surfactants are these kinds where you have what's called zwitterionic surfactants, where they are both positively and negatively charged in solution, but the overall charge on this molecule is 0. So when you put these into an electric field, they don't move. They can coat the protein through these hydrophobic regions. This is a surfactant called C7BZO. And so again, you have the hydrophobic tail that interacts with the protein. You have this head group, which can then interact with water through the positive and the negative charge. But when they coat the protein molecule, the charge on the protein molecule is the charge inherent to the protein because of the side chains of its amino acid. Coating them with this doesn't change that charge, whereas coating it with SDS will give the protein an overall negative charge, which is the reason why it's used in SDS page in one-dimensional gel electrophoresis, where everything has a negative charge and therefore they are getting separated by their mass. In the case of using these zwitterionic surfactants, you're doing isoelectric focusing. You're separating things by the protein's charge. So you don't want the surfactant interfering with that. If you have a protein solution where the proteins are all coated with SDS and you put it onto an isoelectric focusing strip, all the proteins will just focus to pH 9. You won't get any separation whatsoever. So this is just reiterating the same thing where you've got your surfactant molecules and they interact with the external surfaces of the protein, but they can also interact with the internal surfaces of the protein and open them out to be able to get more molecules absorbed. You can go too far. You can have too many surfactant molecules. And when you've got too many surfactant molecules in solution, rather than having protein to be able to interact with and bind to, they start to interact with themselves and they form these micelles in solution. And these can actually interfere with downstream analyses. They can interfere with things like electrophoresis and chromatography, and they can make things worse. So you need to understand what's called the critical micellar concentration of your surfactants and make sure that you don't exceed those when you're doing your extractions, because you'll just make your life worse. Yeah, all of the animations that I haven't used. So breaking these things open is only one of the problems. So yes, you can add chaotropes, you can add surfactants. They won't do much by themselves. So you need to be able to disrupt things a lot better. And the other thing that you want to do is actually massively increase the surface area of the sample to be able to extract the proteins out as efficiently as possible. And there's a couple of different ways of doing this. So you add your chaotropes and you add your surfactants and things like that. One of the things you can do that's very popular with bacterial cells is that you use something like a French press, where the cells are being pushed through a very, very fine orifice that's actually smaller than the size of the cell. So by pushing them through that at tens of thousands of PSI, the cells break open. They burst. You put a hole in them and the contents are able to be released. It's great for releasing soluble protein if you want to do, like if you're doing recombinant expression, to be able to get soluble things out of the inside, but it's not so great if you actually want to do membrane extractions. Better things to do are things like use ultrasonic probes, where the ultrasonic waves you're putting into the liquid sample rattle and shatter the cells, but they also shatter DNA, which is a very good thing when you're trying to do protein extractions. You want to shatter your DNA up so that you don't have these massive DNA molecules that you have to get rid of. The other thing you can do is, if you're dealing with bacterial cells, is you can use things like lysozyme, enzymes that actually break down peptidoglycan and things like that. I'm not a huge fan of these things, and the reason is that I don't necessarily like adding proteins to samples of proteins, because if you add too much of this stuff, all you start to do is start to measure the presence of those molecules. You're already adding trypsin at low concentrations. You're potentially adding benzenase to shear amino acid, to shear DNA. So the less number of things you can actually add, the better. And what we find is that rather than using things like lysozyme, actually snap freezing cells and lyophilizing them to a powder where you remove all of the water out of the sample is a much better way of doing this, because you can then put them in a ball mill and grind them to talcum powder. And because there's no water, these things become very, very brittle, and you can grind these down to very, very, very fine particles, and therefore you've got smaller pieces for the chaotropes and the surfactants to actually interact with and go inside to solubilize the proteins back out of them. So that's the end of my brief explanation of the way that sample preparation works. I hope that's actually been useful. I think the take-home messages of these is that if you can't solubilize your protein, you're not going to be able to analyze it. You're not going to be able to fraction it. You're not going to be able to collect all of the molecules of the same protein together to be able to increase your sensitivity, because that's the most important thing. Proteomics is a concentration sensitive technique. If you can't collect all of your proteins together, your proteoforms together in the smallest volume and therefore the smallest piece of space, you can't analyze it with the greatest amounts of sensitivity. Carefully plan your experiments. Think about what your the additives that you're adding to your sample are going to do. What is this surfactant going to do? Is it going to affect your ability to do trips and digest? Are you going to have to dilute your sample down? Are you going to have to do something crazy to overcome this thing that you've added? One-pot sample preparation is becoming extremely popular, and for very, very good reason, because when you don't have to pipette things out, or you don't have to do multiple stages where you're transferring things around, the chances of you losing protein, the chances of you losing proteoforms is massively reduced. If you're into doing peptide-centric shotgun proteomics, one of the great ways of doing this is this 1SPE method that has been developed by Brian Hampton. I recommend that you go and have a look at his GitHub. The methodology is very, very easy to follow. It's extremely robust. It's very cheap, and it gives some very, very good results in shotgun proteomics. So I hope you've enjoyed this video. If you've got any comments, please leave them in the comments below, or feel free to get onto the University of Technology Sydney website and hunt down my email address and send me an email if you've got any questions, comments, or you want any elaboration.